Taxonomy. Taxonomy is the hierarchical system of identifying organisms, naming them, and classifying them. And while doing this taxonomic classifications, we use phenology or cladistics, phonetics, or ontogeny. There are various processes by which we can do the taxonomic classification of organisms. Hello friends, I am Dr. Ayantika Das, and in this video I am going to give you an overview, a brief overview of phenology, cladistics, phonetics and ontogeny, and alongside I will be solving one set question from Gujarat set. So let's start the video. So the question is, a taxonomic system based on phenotypic similarities equally weighted but without disregard for evolutionary relationship. So the question is that evolutionary relationship is not regarded but the phenotypic similarity is regarded. So if you consider the only phenotypic similarity, what type of classification system do we use? So the options are phylogeny, phonetics, cladistics and ontogeny. So I am going to discuss a brief about all these four, and at the end of the video I will be answering this question. Now phylogeny, it is basically classification based on the evolutionary history. How do we make the evolutionary history? By morphological traits, that is heritable traits, as well as biochemical. DNA sequence, protein sequence, that is the amino acid sequence of the proteins, they are regarded, for example, some protein from mitochondria of two different organisms, they are considered and their amino acid sequence, they are compared so that to determine the genetic relatedness. More the sequence more the sequence is closely related, then more will be the similarity between the amino acid sequence. So closely related species will have similar DNA sequence, more similar DNA sequence than distantly related species. So this phylogeny is based on both observable heritable trait as well as biochemistry. Means morphology and biochemistry, these are the two very important things that are to be considered while we do phylogeny. And in fact, phylogeny, it is not only in classification but also very important in if you consider the biodiversity or the evolutionary history or ecology or we consider the genetics. So phylogeny can be of the phylogenic, these are the phylogenic tree, and we see that this is a unrooted tree. This is an unrooted tree and this is the rooted tree. Now we can easily find out the difference between the two. That is, here in this unrooted tree, we are not sure of the ancestor. In this unrooted phylogenic tree, we are not sure which one is the common ancestor. So we just take a single, say for example, this, if this is the phylogenetic unrooted tree, and if this is A, this is the organism B, say for example this is the organism C and this is the organism D. So then if we just have to hypothesize that maybe A is our ancestor. common ancestor. Then according to it, we will be finding out the relatedness. But here we are hypothesizing. But in this rooted type of phylogenetic tree, we see that there is a common ancestor, and we don't have to hypothesize. And in fact, we see that this is a very branch or tree like system, but this one, it is radial. This one is a form of unbranched. Then we can have this type of branching also on radial, and then we can also have this type of branching, branching also, radial type of branching, or just a Y type of branching. This type of branching can be seen in unrooted phylogenetic tree, but in rooted we find that this type of, and this is the ancestor, and in fact this one is the these are the branches. These are the basal branch, and all these are terminal branches. Here this one, and this one are the terminal branches, while this one is the basal branch, and these are the nodal points. These are the nodal points, and these are the terminal nodes. For example, crocodilians, these are terminal node. And in fact, here we see that the terminal node, they are also called the leaf organisms, the leaf nodes. These the terminal ones are called. And so we see that this one, common ancestor, it is the basal taxon. We can say this is the basal taxon. This one is the basal branch. I'm writing it as BB, basal branch. This one are the terminal branch. And we know that these are the nodes. This is the nodal point, this is the nodal point, this is also a nodal point. We have some ancestor here also, and in fact these are the leaf nodes. These are the terminal point are called the leaf nodes. And so this is pretty much how this is how the phylogram looks. Cladistic is also a process by which we can do taxonomy, but in this cladistics we are considering the evolutionary change, that is from the parental ancestral character to the shared derived character. So the entire relationship, evolutionary relationship, are considered, but not the genomic aspect, that is the biochemical, the protein and the DNA, the level of similarity is not considered here. So the genetic relatedness is not considered. So we see that we are not being able to, if we see a cladogram, we are not able to find the genetic distance or genetic relatedness. We only know that these species are related, but how much they are related, that we are not able to know. So the tree that we see in while doing cladistic is called cladogram, and the tree that we have seen in while doing phylogeny, that is called the phylogram. And here we see these are the examples how cladogram look, and these are few terms that are very important. If you know this, what are these terms, then we will be able to find out the problems. We will be able to solve the problems regarding this. Cladistics. And in fact, cladistics and phylogeny has been repeated multiple times in both CSI net and set questions. So let's see. Here we see in this diagram, we see that this is the ancestral common ancestor, and the common ancestor by repeated nodes, we see that these are the two derived characters. These two organisms, these two species have developed some derived character. These are the ancestral characters. These are the taxa that are bearing the ancestral character, and I'll be drawing it in other color. So these are the ancestral character bearing taxa. We see that they have the same characteristic, and on the other hand, the things that I marked in yellow, so these are the derived characters. So any this type of cladogram where this type of derived character is developed separately by two taxa without any common ancestor. See, there is no common ancestor here. Here there is no common ancestor. So there is no common ancestor that have this derived character. This is the ancestor that these two organisms, these two taxa come from, but they have a different character. That is different from the ancestral character. The ancestral character is the gray colored portion that I marked with red, and the derived character is the black colored. So any cladogram where two individual taxa has developed certain derived character which is absent in their ancestor, then that type of similarity, it is called the apomorphy. And where any taxa, they have the same character as their common ancestor, then this is called the pleisomorphy. So this is apomorphy and this is pleisomorphy. So again we see autapomorphy. Autapomorphy is that when all the organisms, that is the common ancestor, common ancestor I'm writing CA, the recent ancestor, that is I'm writing as RA. So both the common ancestor, recent ancestor, they have the same characteristic, but a derived character is formed in a new taxa that develops into a new taxa. Such type of relationship we can say is autapomorphy. But if this is a common ancestor, this is a distant common ancestor, but I am writing as DCA, and this one I am writing it in green. So this one is the common ancestor, recent common ancestor, recent, that I am writing as RCA. So the recent common ancestor has the same derived character as that of the progeny taxa. So these two taxa, they have the same character as its recent common ancestor, which is different from the distant common ancestor. So this type of relationship, it is called the synapomorphy. On the other hand, here we see in this homoplasy, we see that this is the distant common ancestor that I am writing as DCA. Distant common ancestor have a character, and these two taxa, say for example this is A, this is B, this is C, and this is D. So taxa A and taxa D have the same derived character, but they do not share a recent common ancestor. They do not share a common ancestor, but they have individually, separately developed certain derived character which is similar. So this type of relationship, it is called the homoplasy. So A has the homoplasy with D. So A is homoplastic with the
already
D.
told that there
So
are
this is
certain
about the
difference
cladogram.
between a cladogram and a phylogenetic tree, where here in this example, here we see that the cladogram, the branches are of different length. This is much longer than this one. So this branch length, it depends upon the genetic similarity. So we see that this American no marsupial mole and this wombat, the amount of distance is much smaller than this distance. So the distance determines the genetic similarity. So here in this phylogenetic tree, we see that this distances of the branch distance, that is, they are of different length, and the length of the branch distance is directly proportional to their genetic distance. More the genetic relatedness, lesser will be this distance. So but this is the case of phylogenetic tree, that is, they have the distance of each branch is proportional to the amount of inferred evolutionary change. But in case of cladogram, it is bit different. Here in this cladogram, we see that all they are almost, they have this is a slanted cladogram. We see that the genetic distance is same, which means that this cladogram, they put importance on the relatedness, but the how much is the relatedness, how much is the genetic similarity, we are unable to find through the cladogram. So from this point of view, we can say that phylogenetic tree is much more informative than the cladogram tree. Then we come to phonetics. Now phonetics is mostly, it is mostly based on the number of traits that are similar. That is, number of morphological traits that are similar. More the number of traits that are similar between two organisms, then more will be the accuracy by which we can relate or disrelate these two organisms, which means that we will require a large amount of data set, because we put this data in a software and we do cluster analysis. And while we do cluster analysis, we know that in cluster analysis, more accuracy is obtained if the data set is huge. That is, more the number of similarity between two organisms, more will be its relatedness or closeness. But in phonetics, we consider the only morphological external characters. We do not consider the genetic relatedness. It is just done by software, and it is also interchangeably used. It is closely related to numerical taxonomy and often interchangeably used as numerical taxonomy, though many taxonomists consider it a bit different from phonetics. Phonetics is a bit different from that of numerical taxonomy. But whatever be the difference, we both use the data, that is phonetics and numerical taxonomy. We use this data based on the external morphological similarity. But this external morphological similarity can arise due to convergent evolution also. So then that will be a problem, because we two the two taxa that has evolved by a convergent evolution can show similarity in morphology and body structure, but they are not closely related. This is a convergent evolution. They are converging towards a character, but they are not that much closely related. So then it becomes a bit confusing also. Then about the ontogeny. Now we know that ontogeny is about the origination and development. That is right from the formation of a zygote till the formation of an individual, all the developmental process, that is the formation from uni to multicellular, then tissue formation, organogenesis, all the entire process, the developmental stages, they are considered while considering the genetic relatedness or evolutionary relatedness. How it is done? This is because of evolutionary developmental recapitulation theory. Now, by this recapitulation theory, the very common example is that all the birds and even the human, they have some certain characters which is similar to that of the fish. That is, during our developmental process from a zygote to an entire organism, we at certain stages, also a human as well as the bird embryo, they show the gill slits. But gill slits are absent in adult bird or adult human being or adult mammals. But still, in our developmental process, we are seeing at certain stage we are having gill slits, which indicate that our common ancestor of both birds and that of the mammal is fish. So this is a very common example of how the recapitulation theory, how ontogeny, by studying the ontogenesis, that is the developmental biology, we can easily find out the genetic relatedness. The evolutionary relatedness. Even distantly related species can be found out by the help of this ontogeny. But ontogeny is a bit different from that of phylogeny, because in ontogeny we are finding the developmental history of a single individual species. But in phylogeny, we are relating the evolutionary history of different species. So now we come to our question, that is a taxonomic system that is based on phenotypic similarity, equally weighted and without regard to the evolutionary relationship. Now let us consider our options. So in phylogeny, we consider the evolutionary relationship as well as the biochemical relationship. That is evolutionary relationship both in morphology as well as the protein and the DNA sequences. So our answer phylogeny is not correct, because in phylogeny we not only considered the phenotypic similarity but also the evolutionary similarity. So in both these cases are considered, so phylogeny will not be the answer. Next we see cladistics. Now in cladistics, what we do is that we find out the derived characters, then by this we find out the evolutionary relationship. So cladistic will also not be the option. Then in ontogeny. Ontogeny is the developmental biology, that is from the cell division to the embryogenesis and all. So in developmental biology, what we find is that by studying the developmental biology of different species, we find the evolutionary relationship. Here also we are considering the evolutionary relationship. So ontogeny will also not be the answer. But in phonetics alone, we are saying that we are considering similarity between the phenotypic similarity, that is the external similarity, all the external characteristics are considered without any regard of the evolutionary relationship. So our answer will be option B, that is phonetics. which is also interchangeably used as numerical taxonomy. So that was all about the question solving and an overview of phylogeny, cladistics, phonetics and ontogeny. Hope you like this video. If you like this video, please do hit the like button and share it with your friends so that it gets maximum views. Please subscribe to my channel to get more such videos and also to encourage me. Those who have already subscribed, thanks for subscription, and I make videos on CSI and NET and SET as well as MSc and BSc syllabus. You can go to my channel and watch those videos. I've already made videos on algae, fungi and my lichens, cyanobacteria, bryophyte, pteridophytes and on CSI and NET as well as SET videos. And thanks for watching this video.